Assalamualaikum and hi everyone. So our group will be presenting about biodiversity in helminths and nematodes as a field of study. So before that, let me introduce myself first. So my name is Nur Shifa binti Muhammad Sabri and this is all our members. Nur Najwa, Nur Alfa and also Maria Elsa. So later, when they start presenting their topic, they will introduce themselves. So for this presentation, we will start by understanding what helminths actually are. So the definition of helminths. Helminths is a general term meaning worm. The helminths are invertebrate, characterized by elongated, flat or round bodies. In medically oriented schemes, the flatworms or platyhelminths, platy from the Greek, root meaning flat, include flukes and tapeworms. Roundworms are nematodes, nemato from the Greek word meaning track. So in simple term, it is a group of parasitic worms that can live inside of humans, animals and sometimes plants. They survive by obtaining nutrients from their host, and many of them can cause diseases or reduce the health of the host. So this is all the major groups of helminths. Helminths are divided into 4 major groups, which are Tuberlaria, flatworms, Cestoda, tapeworms, Nematoda, flukes, and also monogeny. And then next, I will explain about the definition of nematodes also. Nematodes are a type of helminths that has a round, cylindrical, and non-segmented body. They are one of the most diverse and abundant organisms on Earth. Nematodes can be either free-living in soil, freshwater, or marine environments, or parasitic in humans, livestock, or plants. Their diversity is so high that more than 25,000 species have been described, and scientists believe that there are hundreds of thousands more of these species. When we talk about biodiversity in helminths and nematodes, we are referring to the variety that exists within this group. So, the definition of biodiversity in helminths. Biodiversity in helminths refers to the variety and differences found within parasitic worms, including their species, physical structures, life cycles, host range, and ecological roles. So, in other words, it describes how diverse helminths are in terms of variety in species, in morphology, in life cycles, host range, and also ecological niche. And next, I will present about explanation of the 4 helminths group. So first, I will start with Tuberlaria, flatworms. They have a soft, flat body covered with cilia that allows smooth gliding movement. So for information, cilia is like their kind of feet. Small fish. Okay, and their size varies widely from microscopic to several centimeters. They are free-living and commonly found in freshwater, marine areas, or moist land. So Tuberlarians are active predators or scavengers, feeding on small organisms or organic materials. So this is like their diagram, their body diagram, if you want to know a lot more about Tuberlaria. Next, I will explain about Cestoda, tapeworms. They appear flat, ribbon-like, and segmented with a head that attach firmly to the intestine. Some species can grow extremely long, reaching several meters. They live inside the intestine of vertebrates as internal parasites. Tapeworms absorb nutrients directly through their body surface. From this diagram, you can actually say that tapeworms really look like a ribbon, their body. So next, Nematoda, flukes. They have a leaf-shaped flattened body with 2 suckers for attachment. Most species are small to medium, usually only a few millimeters to a few centimeters. They inhabit organs like the liver, lungs, and blood vessels of humans and animals. Flukes remain attached to tissues and feed on blood or body fluids. So this is what they look like. Okay, next, I will explain about monogenes. So they are small flatworms with a strong attachment organ called an opisthecter. Their size is generally between 1 to 20 mm. They live on the gills, skin, or fins of fish. Monogenes cling tightly and feed on mucus, skin cells, or blood. So this is what they look like. Then I will explain about nematodes, roundworms. They have a long cylindrical body with a tough outer cuticle for protection. Their size range from microscopic forms to several centimeter adults. They can live in soil, water, or inside humans, animals, and plants. Nematodes move actively and feed on organic matter or nutrient from the host.
Assalamualaikum and hi, my name is Nur Najwa and I will explain about the morphological diversity in helminths. Morphological diversity refers to the wide variation in the physical structures of helminths. This includes differences in body shape, body plan, and organ system, which help us understand how diverse each group is. Morphology is very important because it reflects evolutionary adaptation. It shows how helminths survive in different hosts, attach to tissues, and complete their life cycles. It also helps in classification and identification, especially during laboratory diagnosis. Helminths show major differences between flatworms and roundworms. For example, nematodes like Fasciola hepatica have a flat, leaf-shaped body with strong oral and ventral suckers. These suckers allow them to attach firmly inside organs like the bile ducts, and their flattened body helps them absorb nutrients easily. Next, cestodes like Taenia saginata look very different. They have a long, ribbon-like segmented body made of proglottids. At the front, they have a scolex with hooks and suckers, which help anchor them to the intestinal wall. And their segmented body also allow them to produce egg continuously. Meanwhile, nematodes like Ascaris lumbricoides are cylindrical and unsegmented. with a tough outer cuticle that protects them from digestive enzymes. Their simple, elongated shape helps them move easily inside the intestine. Overall, these structural differences show how each group has evolved unique features to survive in different environments and hosts.
Assalamualaikum and hi, my name is Narafa and I'm going to explain about the life cycle of helminths and nematodes. Helminths has complex and varied life cycle which is part of their biodiversity. For the life cycle, they have two types of life cycle, which is direct life cycle and indirect life cycle. So first, let's take a look at the figure for the direct life cycle. This is example for Ascaris species from phylum Nematodes. First, the adult Ascaris nematodes, the round worm, live in the lumen of a small intestine, and then the female worm may produce approximately 200,000 eggs per day, and the eggs will excrete with the feces. The infertile eggs may be indigested but are not infective. After the female lay an eggs, in the environment, the fertile eggs, depending on the condition, embryonate and become effective after 18 days to several weeks, and the optimal condition are moist, warm, and shaded soil. And then the effective eggs are swollen by a human. In the human body, in the small intestine, the eggs will hatch into a larvae. And then the larvae will penetrate the wall of the small intestine, and they will migrate via the portal circulation through the liver, then via the system circulation to the lungs, where they mature further, which is between 10 to 14 days. And then after they migrate, the larvae will penetrate the alveolar walls, ascend the bronchial tree to the throat, and are swollen. They are returned to a small intestine where they develop into adult worms. Okay, for the next life cycle is indirect life cycle. Indirect life cycle, it require intermediate hosts, for example like snail, insect, or fish. Okay, let's take a look at the figure. First, the adult nematodes, the roundworm of Acanthoensis, live in the pulmonary arteries of rat. The female worm will lay eggs that hatches into the larvae, first stage. Then the first stage larvae will migrate to the pharynx, are swallowed, and are passed in the feces. The first stage larvae penetrate or are ingested by an intermediate host, which is snail or slug, and develop into the third stage larvae. When an intermediate host is ingested by the definitive host, the third stage larvae migrate to the brain, where they develop into a young adult. The young adult return to the venous system and then to the pulmonary arteries, where they mature. Human can acquire the infection by eating a raw or undercooked infected snail or slug. Raw produce that contain a small snake or slug, or part of one, or a transport host such as certain land crab, frogs, toads, or freshwater prawns and shrimps. In human, the larvae will migrate from the gastrointestinal tract to the meninges. Occasionally, they invade the eyes or the lungs. Acanthoensis do not reach full maturity in human. Okay, this is three types transmission modes for helminths. What is transmission modes? Transmission modes is refer to how a disease or infection spread from one host to another. Okay, for the helminths, there are three types, which is oral, skin penetration, and vector bonds. For the orals, the host become infected by eating effective stage like eggs or larvae in contaminated food, water, or intermediate host. For the skin penetration, the larvae penetrate the host skin directly, usually from contaminated soil or water. And the vector bonds transmission, the infection occurs when an arthropod or insect intermediate host carries the parasite to the definitive host, like human.
Next, I will talk about ecological diversity in helminths. Helminths also exhibit ecological diversity, which means they can live in a variety of environments and habitats. Their ecology is closely linked to their life cycle, mode of transmission, and the type of host they infect. Some helminths depend on aquatic habitats, while others rely on soil or internal tissues within their host. This variety of habitats show just how adaptable and diverse helminths are. For example, Fasciola hepatica, which is a trematode, is commonly found in the bile ducts of mammals such as cattle and humans. Its juvenile stage depend on freshwater environment where they infect snails before emerging onto aquatic plants. This shows how trematodes rely on both aquatic and internal host ecosystem. Next, cestodes like Tania saginata live in the human intestine as adult, but their larval stage develop inside the muscle of cattle. This demonstrates an ecology that involves two completely different hosts and two distinct habitats: the intestinal environment and animal tissue. Meanwhile, nematodes like Ascaris lumbricoids live in the human small intestine, but their eggs survive in contaminated soil, where they can remain viable for long periods. This makes soil a very important ecological component in their transmission. Overall, helminths show ecological diversity by occupying different environments throughout their life cycle, from water and soil to various organs within their host. This diversity allows them to adapt and survive under different conditions.
Okay, next, methods of study biodiversity in helminths. This is the tools that scientists use to study biodiversity in helminths. There's a lot of tools, but this is the tool that usually they use. For the first one is integrative taxonomy, which is combined morphology, molecular, ecological data, and cryptic species identification, standard best practice. For the second, they use tools and techniques, which is morphology. They use microscope staining and slide prep, PCR, DNA sequencing, and barcoding. And the last one is application. They use diversity surveys, ecology and epidemiology, and populations genomics.
Okay, importance of studying helminths and nematode biodiversity. The first one is supports public health improvement. By understanding helminths and nematodes helps identify disease-causing species, improving prevention, diagnosis, and treatment strategies. Enhance disease control programs. Biodiversity studies reveal transmission patterns, host relationships, and environmental factors that influence parasite spread. The third one is guides environmental and ecosystem monitoring. Many nematodes act as bioindicators. Their presence and diversity signal soil health, pollution level, and ecosystem stability. Next is promote sustainable agriculture. Identifying harmful and beneficial nematodes help protect crops, improve soil quality, and guide pest management strategies. Advanced scientific knowledge and taxonomy. Biodiversity research leads to new species discoveries and a better understanding of evolutionary relationship. The second last one is supports biodiversity conservation efforts. Parasites are part of natural ecosystem. Studying them help maintain ecological balance and understand species interaction. And the last one is improve One Health approaches. Integrates human, animal, and environmental health perspective by examining helminths that cross special species barriers. Issues and challenges by expert. The first one is morphology and identification. It is high similarity in physical features, makes it difficult to distinguish between species, often causing misidentification. Many species look identical externally despite being genetically different, known as cryptic species. The second one is host and habitat. Some species infect multiple hosts, making it challenging to map host parasite diversity clearly. Sampling extreme environments like deep soil layers or marine hosts is limited due to cost and accessibility. The third one is data and research limitation. Genetic reference databases for helminth and nematodes are still incomplete, slowing species verification. The overall biodiversity is underestimated because many species have not been fully discovered or studied, especially in certain regions. The fourth one is environmental change. Climate change shifts species distribution into new areas, disrupting biodiversity records and tracking. Increasing drought-resistant nematodes make ecological and control research more complicated. And the last one is expertise and technology. Helminths taxonomy has fewer specialists as it is a highly niche field with limited experts. Advanced molecular and sequencing tools are more reliable than microscopy, but they remain expensive and require skill training. So we have come to the end of this presentation. So this is all the points that we have learned from this topic. So first, reveals the wide range of sizes, forms and adaptations of helminths and nematodes. Next, highlights their roles in ecosystems, including effects on host and nutrient cycles. Moreover, provide insight for research in parasitology, evolution, medicine and agriculture. So that's all from us, I think. Thank you for listening.